Hello, everyone, and welcome. We're glad you've joined us for this live webinar, Laboratory Automation to Deliver Reliable Results Faster, presented by Dr. Stefan Overkamp. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. This webinar is sponsored by Beckman Coulter. Beckman Coulter is dedicated to advancing and optimizing the laboratory. The company's global leadership and world-class service and support delivers sophisticated instrument systems, reagents, and services to life science researchers in academic and commercial laboratories, enabling new discoveries in biology-based research and development. For more information, please visit www.beckman.com. Now let's get started. The seminar is designed to be interactive. You can submit questions to the speaker at any time during the presentation. To do so, simply click the green Q&A button at the lower left of the presentation window and type them into the Q&A box. Questions will be answered after the presentation, and we will answer as many questions as we have time for. If by chance we weren't able to get to your question will be answered via email following the presentation. You will be viewing the presentation in the slide window. To enlarge the window, click on the screen icon in the lower right-hand corner of the slide window. If you experience technical problems seeing or hearing the presentation, just click on the support button found at the top right of the presentation window, or report your problem by clicking on the green Q&A button at lower left. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credits. Our speaker today is Dr. Stefan Overkamp, Senior Field Marketing Manager for Beckman Coulter Life Sciences. Dr. Stefan Overkamp has over 15 years of automation, cell culture, and molecular biology experience. He received his PhD in molecular biology from the University of Munster, where he trained with a focus on biochemistry and molecular biology of plant pathogen defense. I will now turn my presentation over to Dr. Stefan Overkamp. Dr. Overkamp?
Yes, thank you, Christina, and welcome everyone. I'd like to thank LabRoots for giving me the opportunity to speak here about laboratory automation and how that enables to deliver reliable results faster. If we look at the agenda, I want to start with a brief introduction on laboratory automation, how we achieve higher throughput through parallel processing, how we achieve more efficiency through longer hands-off time, and how we deliver reliable data through increased reproducibility and integrated data handling. After this introduction, I want to give you a couple of examples from two fields of application, one from the genomics workflows and one from cellular workflows. And in the end, after the summary, we have some time for a Q&A session, and I'm looking forward to your questions on laboratory automation. So for the intro, how do we achieve higher throughput? Well, the key to higher throughput usually is parallel processing. And if you look at how you handle samples in your laboratory at the bench, it's usually performed using single-channel or sometimes 8-channel pipetters. All of that is available in laboratory automation as well, but on top of that, laboratory automation does offer the user a higher degree of parallel processing with, for example, 96 or even 384-channel heads. When it comes to high throughput or even ultra-high throughput applications such as screening, for example, or high throughput genomic extractions, these systems enable a significant speed advantage. And I'm just giving you a very simple example down here, which is filling a 384 well plate with 10 microliters of a buffer from a reservoir, which takes, using a single channel, approximately 52 minutes, while using an 8-channel, you're already down to 8 minutes. If you use a 96-channel, you're down to a good 1 minute, and with a 384 channel, it's below 1 minute. You can actually see here that at the 384 channel head usage, this is the most of the time is not the actual pipetting, because the difference between the 96 and the 384 is so small. But the most of the time is used for loading the tips, actually, because this scenario is done with disposable tips. So if you have multiples of these plates instead of filling a single one, the advantage would be even much more significant. Where does this give you a real benefit? Well, there are certain types of applications that are usually requiring lots of pipetting steps and have a lot of samples at the same types. And typical applications would be ELISAs, cell-based assays, cell staining assays, typical biochemical assays, compound management, serial dilutions, plate replications, high-throughput DNA and RNA extractions, and high-throughput next-generation library preparations. Also, laboratory automation offers more efficiency through longer hands-off time. So the classical work at the lab bench does require the researcher, well, actually, to be at the lab to perform this research and this pipetting. Automation systems of different levels of complexity actually do allow to automate full workflows instead of only the liquid handling itself or processes or processing of multiple plates. Because liquid handling systems of today, they cannot just do pipetting, they can also store labware supply, reagent supply, they can move labware around, trigger integrated devices for such processes, such as thermocyclers or readers, and the advanced software methods, they will execute the pipetting and other tasks to complete a full method successfully. And when I say full method, for example, here I'm looking at an NGS library preparation, where if you do all of that manually, you can probably generate up to 8 libraries with a full day of hands-on time manually. But in an automated fashion, we are able to generate 96 libraries with less than 2 hours hands-on time. So you're not just getting more libraries, you're also freeing up 6 hours of your working time to do more interesting work, for example, interpreting these data and drawing scientific conclusions from these data. Very important is also that the data you are generating in your experiments are reliable, that they actually do reflect biology rather than experimental conditions. So automation helps generating reliable data in increasing reproducibility, and it also helps by allowing for integrated data handling. Automated liquid handling or automated laboratory processing is controlled by computers, and the Biomek automation control software is built on a database. And what that means is that data handling basically is in its DNA. So whenever in Biomek we do pipette liquids, we also virtually pipette the data. And at the image to the right, you can see that these data can be a lot of things, not just sample ID, but concentrations or the actual protocol you are running, the source plate, a batch number, etc., etc. On top of that, automated liquid handlers also drive reproducibility day in and day out. They are a machine, right? They do not have a bad Monday after an exciting weekend or anything like that. They can work overnight. Usually this effect, again, plays if you are looking at thousands or even tens of thousands of samples more efficiently versus if you're looking at 8 or 12 samples. I mean, a very experienced TA can get very good results with respect to reproducibility if that person does prepare, let's say, 8 or 16 NGS libraries. But if you got to prepare 96 or 384 of these libraries, usually the laboratory automation approach gives you a much higher degree of reproducibility. Okay, so that was just a brief introduction, and I'd like to now continue with some examples of genomic workflows. So I've picked NextGen library as an example. And before I go into details, I just want to give you a high-level overview of what a typical NGS workflow is. So basically, it can be broken down into these parts that you do see here. If your starting material is RNA, you would start with a cDNA synthesis upfront to turn RNA into DNA. Then the actual library construction does happen. And optional, you can enrich that library by applying PCR, and you can limit the segment of the nucleic acid you are looking at by doing a target capture. And then finally, you have the library QC and the actual sequencing happen. The thing about NGS sample preparation or NGS library construction is that the specific workflow, dependent on your kit that you are using and the sample material and your sequencing interest, it can vary greatly. It can be a single day, short and simple, but it can also be multi-day, highly complex protocols. Common elements of such a workflow are reagent and enzyme transfers, sample transfers, bead-based sample cleanup and or size selection, and thermal cycling and incubations. And if we look at a couple of examples just on how complex these are, I have 3 examples. 2 you see here. To the left, you have the Rubicon Genomics Throughplex Plasma-seq. That is a process that gives you a library in approximately 3.5 hours. It uses a single plate for all reactions, but it has a limited number of start and stop points. To the right, you see a workflow of the Illumina TrueSeq Nano DNA protocol. This protocol takes already a full day. It has multiple reaction and cleanup plates and multiple start and stop points. But we can go even further. So what we see here is another kit from Illumina, the TrueSeq RNA Access. That takes full 3 days to give you a library. It includes target capture. It includes large number of labware and consumables. And it is a very complex workflow with numerous start and stop points. So you can see NGS library prep, even though high level it's pretty simple, if you're going down to the detail, it can be very complex. And variability is significant. So what are the challenges of manual NGS sample preparation? Well, first of all, there is a pretty significant amount of manual pipetting steps. So there's high variability. So for example, inefficient adapter cleanup can occur. The sample yield can be very different and vary significantly. It does require skilled pipetting. For example, the accurate transfer of viscous liquids, the in-well tip mixing, and the preventing of the carryover of magnetic beads. And because of that, and because it takes such a long hands-on period, it is prone to user fatigue and injury. By that, it is easy that users do introduce errors in that process. For example, sample or index transfer mix-ups, sample normalization errors, master mix receipt calculation errors, pipette volume settings errors. And overall, the throughput is limited, and that means you're facing reduced data generation. Limited personnel resources, because you could do parallel processing, as I mentioned before, by not using automation, but using multiple TAs, but usually that is too costly and those people are not available. And there's more effort spent on routine sampling processing versus the actual data analysis, which usually is the more exciting part of science. Automation can help to minimize or even eliminate these challenges. And here's a couple of examples. No, we start with some things we hear in our everyday work talking to customers. A lot of our customers, when we approach them about automation, give us a response that they're saying, hey, we're too small for automation. Well, you're never too small for automation, because automation is not necessarily only targeted at high throughput. Automation does reduce the effort of routine processing and does increase productivity even with limited personnel resources. If you have one person that does actually understand to run a Biomek or a liquid handler, that already gives you a significant increase in efficiency. We also often hear that the throughput would not be high enough to justify automation. Again, the key point to adapt automation is more on the accuracy and reproducibility of sample data, because that is what matters in science, right? If you do not have good data, if you don't have reproducible and accurate data, any scientific conclusion is flawed or is possibly not correct. Also hear that people do trust people more than machines. Well, what we deliver, especially in the context of NGS, is not just a liquid handler. We do know that these methods do need to be qualified by knowledgeable people, and we're cooperating with the kit vendors and with some key users in the field to demonstrate and do actually qualify with data our methods that we do deliver. We also hear that the budgets are limited. Well, sure, in the end, we will send an invoice and ask for some money. But automation overall can provide significant savings over manual processing through reducing the labor cost and the overtime pay, reduce the rework that is caused by human error, and actually reduce the consumable usage by using efficient labware and tip handling. And the final objective we often hear is we don't have the expertise to operate and maintain laboratory automation. And this is an important one, I have to say, because if you look around in the field, you can see a lot of automation systems that are no longer used for that reason. The Biomek software is a full-featured but easy to use, which makes it simple for the right solution to be executed. And on top of that, and I'll get to that in a couple of slides, we are offering what we call the Biomek Method Launcher, a very simplistic user interface. So if a user has no intent to modify or change any of the methods, it's basically bringing the system down to a push-button solution. Also, for different types of requirements with respect to throughput, with availability of budget, we can offer different platforms that enable the automation of such processes as NGS sample prep. To the left, you do see our entry-level liquid handler, which is the Biomek 4000. The size is just right for low to medium throughput labs. So let's say if you're producing 1 to 24 libraries at a time. It does offer a pod with interchangeable pipetting tools, single and 8 channel, which gives you full flexibility with respect to the number of samples you want to process. So our protocols do not force you to use 8 or 16 or 24 samples at the same time, but you can have continuously any number of samples being processed on that system. It comes with an optional enclosure that allows you to protect your samples from any interference of airborne particles from the laboratory. To the right, you see the Biomek NX. It is for medium to high throughput, up to 96. It comes in 2 variations, one with a span 8, one with a multi-channel, and that really depends what is more important to you, flexibility or throughput. And it has 16 positions, high customizable deck with increased capacity. The final one, and the high-end solution, is the Biomek FX platform. It is high throughput, complex workflows, and can process up to 96 samples at the same time. It can be configured with a combination of 2 pods, a multi-channel, and a span 8, so combining flexibility and high throughput. And has an even larger deck with more than 20 positions that allows us to store the labware required for really complex workflows. Okay, so what else is important if we're looking at NGS workflows? Well, it's definitely about accessories and integrated device options. Labware selection, heating, cooling, orbital shaker, tip mixing, tip usage. If we look at that, Labware selection, you want to make a decision if you're using tubes versus plates, what type of reagent reservoirs are required, magnetic plates and bead separation, and thermal cycler lids, which is an important part if you are looking into integrated thermal cycling. Shaking. So it is a fast and efficient plate mixing for mostly bead separations. It reduces tip usage, and it provides very uniform results. On the other hand, tip mixing works very well for large volumes. It does prevent the cross-contamination with labware that is filled at or near the well capacity, and you can access tubes or labware not compatible with orbital shakers on deck. Heating, cooling. So there's two options basically. You can have a heater or cooler with a Peltier that has no sealing or does not require any sealing, but it has a low ramping and cooling speed, and it's not ideal for all applications, especially with higher temperatures. On the other hand, your other options is a thermal cycler. It can be integrated on all platforms that I presented to you a couple of slides ago. It does provide true walk-away solution because it runs its own protocol. It prevents evaporation with plate sealing lids, and it is compatible with common use PCR labware. Also important is the tip type, and you got to make decisions on the various volume ranges, whether you want to use sterile with barrier or non-barrier, whether you want to use conductive or non-conductive for liquid level sensing, and if you do need tube access and what these tube sizes would be. So you also have a couple of options on the actual method workflow. So for example, you can decide whether you want to do on-deck or off-deck thermal cycling. So even though we do have the option to integrate thermal cyclers, we can definitely offer integrating just by the workflow off-deck thermal cycling. You can work in a training mode. That means you can only run the protocol with water, basically, no samples, just to test it. You can have all relevant application-specific options being chosen. You have the option validation, and you have safe stop points. For example, that are points where you can safely store the intermediate product. You can have post PCR break points, and these storage points also do allow for off-deck incubations, and they allow for recovery. Also important is how you handle chemistry on deck. For example, in this case, it's NaOH, which is present in some hybridization and capture NGS methods, such as the Illumina Nextera rapid capture. It is used to denature the captured library and release it from the streptavidine beads. It can also absorb CO2 from the atmosphere if it's set open on the deck for an extended period of time and thus change pH. So it's important to protect that, for example. So now down to a real example of what we actually do. I'm focusing here on DNA sequencing and specifically on whole genome sequencing. Why would you do whole genome sequencing? Well, it's basically used for characterization of genetic variations, mutations across the organisms, de novo sequencing, resequencing, SNP variants, etc., etc. So there's two ways to get to libraries. One's the whole genome sequencing library preparation, as you see it here. You start with a fragment or fragmentation. You do an end repair, an A-tailing, adapter ligation, and PCR enrichment. The purpose is to obtain as much of the genome as possible. Generally, genomes at 20 times coverage are considered finished and can be used for molecular biology applications. There is a different way of doing this, which is the tegmentation, which uses transposons, which basically gives you the same thing, a fragmented library that is good for sequencing purposes. The challenges with whole genome sequencing is you do need to accommodate a wide range of sample inputs. You also need the ability to accurately size select across a wide range of fragment sizes. And a lot of these protocols are complex and lengthy. and they require extensive hand-on time and sample manipulation. What you see here is one example of the TrueSeq DNA PCR-free nano DNA kit from Illumina. It's automated on the entry-level platform, the Biomek 4000. It's also automated on other platforms. It does combine two sequencing chemistry into a one streamlined method, and it does accommodate a range of 100 nanogram to 2 microgram input range. And you can see these workflow diagrams here to the right, where basically the TrueSeq DNA-free, TrueSeq DNA PCR-free, comprises these first two columns, if you wish, and the TrueSeq Nano DNA includes this third with a PCR amplification and PCR cleanup as well. If you look at how we handle this with respect to interacting between the user and the actual sample method, you can see that the Biomek 4000 does provide similar method options flexibility as NX and FX. It's optimized for low input and throughput laboratories. It's scalable to 96 sample workflows if you want to increase from the B4K to another platform. So this is what it looks like. We have a set of tools, single and 8-channel pipetting tools, to provide a balance of precious reagent delivery and efficient cleanups. We use only 3 top tip boxes for the whole process. We have the reagents controlled at 4 degrees Celsius, and the deck is flexible enough to allow for a thermocycler integration, which is not shown here, if that should be required. This is some results we're getting with respect to yield and purity. So these are bioanalyzer data that are produced, and you can see that these are very, very clean, and that the yield is definitely high enough to generate good sequencing results. Total runtime here on the B4K is for 8 samples, 7.5 hours, for 48 samples, 8 hours, and for 96 samples, 9.5 hours. So you can see with an increased number of samples, the efficiency that laboratory automation gives to sample processing really pays off. There is another process we're looking at, which is Swift Biosciences, the Axel NGS 2S Plus. It does provide a PCR-free or optional PCR workflow. We have on-deck thermal cycling optional, and we can generate 96 PCR libraries in approximately 4.5 hours. Again, this is the interface that gives you a lot of options to select from. For example, it's whether or not you want to do PCR or PCR enrichment. We will definitely adjust the spray bead ratios with respect to the sample and input, and so on and so forth. If we look at the results getting from that, you see that the samples we used in this test will generate sufficient library for sequencing, good sequencing results, and these are sequencing results that you're seeing. We have variable fragment length that we can produce. We have a wide range of sample inputs from 100 picogram to 500 nanogram. We have an extremely low PCR duplicate rate and a very good coverage of the sequence. And this is for FFPE samples, and you can basically see the same thing, that the FFPE sample material does produce sufficient yield for sequencing, and again gives extremely low PCR duplicates. These are only 2 examples. A full overview of the methods available in library prep you will find in a method card, and that should be available for download for you today if you click the blue arrow. That gives you access to some PDF files, some more information, detailed information I put together for you. But what about the user experience? So, are you sure we picked the right project? Am I using the most recent version of my method? Or normal Biomek operator is on vacation, and my PI wants me to run this assay. So what do I do? So basically, we introduced a second level of operation. This one here is the standard Biomek software that's basically designed for you to be able to design and modify methods. But very often you don't need that. So if you only want to execute method, we've introduced the Biomek method launcher, and this is the interface that you're seeing. So it will easily allow you to find the correct method to your left, or if needed, via manual control, bring the liquid handler in a state that it can execute the method. So with Biomek Method Launcher, you get a very easy interface that gives you an insight that you have selected the correct method of choice, the number of samples, and some variations that you can easily input here. It also has a guided labware setup, making sure that all pieces of labware that you need to put on the deck are taken care of in the correct order. And when you finally run the system, it has a very nice runtime interface that gives you an up-to-date view on the status of the actual method execution. At the same time, we're taking care of data handling. And again, this you've seen before. There's lots of data, and we call them data sets, that can be associated to a sample or a well, which is not just the sample ID. It could be a project ID. It could be the PI. It could be the species of the sample, and so on. The volume, reagent lot, timestamps, labware type, liquid type, etc., etc. All of that can be handled and tracked and used automatically in the software that we provide with our liquid handling systems. Samples or sample data do come from multiple sources in such a process. So for example, they come from barcode reading of samples, single tubes, from Excel files, etc., etc. So the first step usually is we merge sample data to plate, recording everything. And then in the process, for example, readers will generate data for normalization, if you wish. We need to merge the detector data, track the data and the sample ID to the dilution plate. Then we merge the sample and lot with more process data as we use different types of reagent. And then finally, we need to export these data to Illumina Experiment Manager on a sample sheet, or export these data to a LIMS for downstream analysis. The tool we're providing is called DART, the Data Acquisition and Repository Tool, and that allows to store and merge data from different sources. It's running on a separate database, so you can run it on a separate computer, giving you security on the data that they are safely stored. It can also be viewed on mobile devices, and it can connect to LIMS systems. What you can do is you have the data query builder that does allow you to get an insight into these data based on labware, based on sample, based on run, that gives you an immediate and live overview of the data that are currently in the database on any piece of labware. And you have also the option to generate reports. Here's the reports builder to your right. And above you see a report that is ready to be imported by the Illumina Experiment Manager. So you can modify the reports just so that they are easily connected to other devices or other data handling units in your laboratory. That was what I wanted to present on genomics, and we're now moving on to cell culture. Cell culture has specific challenges, and the reason for that is that cells are just living things. They have specific needs to continue to live and grow happily. While in genomics we're usually working with chemistry or molecules, well, here cells need to be treated differently. They require very gentle pipetting. They do need appropriate room to grow. They need sufficient nutrients. They need appropriate growth conditions. And the cell processes are different in a way that they usually take multiple days, often weeks, and sometimes even months to complete. You have longer periods of incubation, interrupting actual liquid handling tasks. And each individual cell line or clone may grow at a different rate and have different requirements with respect to the media. So let's talk about cell culture process control. Cell culture is a process rather than a method to execute. The challenge is it is often required to automate a full process that does consist of multiple tasks, and they are broken up and separated by longer periods of incubation. Standard liquid handling control software or even schedulers do struggle to handle such lengthy and complex processes. We have introduced a new layer taking care of this, which is called SAMI process management, and that does allow the user to build and manage their own processes, to run multiple processes in parallel on one single system, to manage the system capacity, and manage all data handling for all labware and samples. And I want to give you an example, and here's the example context, which is a real system. This real system consists of a liquid handler, which in this case is the Biomek FX. It's got a couple of components that we do require for facilitating the actions of the process, such as tilting and knocking, incubations, sterility, cell counting, confluency measurements, and transportation and incubation of these plates. If you look at the layout, this is the actual layout of the system, how it's all put together. To the left here, you see the actual liquid handler, the Biomek FX, and then all the other devices that we've just seen in the previous slide are integrated. So that means we can move plates with this SCARA arm across the system. We can bring them from one station to another, execute whatever we need to execute, for example, readout of a confluency measurement here at the Celigo, and then have them controlled in an integrated way so they work together as a workstation that allows us to process plates with all devices as needed. So that was the hardware context. And the context of the real scenario here is this is the process we're trying to automate. We have a manual start. We're starting with frozen cell lines. These are then seeded, and then these cells go on the system, and we continuously maintain these cells. So we measure the confluency. Based on those data, we do media exchange or splitting the cells. And this is happening continuously for 40 different cell lines in parallel. At the same time, the user is able to order cells for their experiments, and if that order comes in, instead of just splitting the cells, we will then upscale the cell number, so increasing the number of cells, and harvesting the cells and make them available for experiments in 96 or 384 well plates per cell line per order request. And here is a breakdown of one process, which is splitting and passaging the cells. So basically what you got to do here is you got to remove the supernatant, you got to wash it, you got to add trypsin, you got to separate the cells, you count the cells, you dilute on a new plate, and discard the supernatant of all plates. Each one of these steps is a single method on a liquid handler. Okay. So how do we integrate that to a full process as you've seen in the previous slide, high level? Well, what you do is down here, you have your liquid handler methods. They are then together make a SAMI method, a scheduled method that takes multiple of these liquid handling actions into a method, and then multiple of these methods like media exchange and passaging make a process that is defined and managed in SAMI process management, and data handling in the background, just as we've seen in the NGS field, is taking care of all the data handling in the background. So it's really bottom up, taking care of all the details, but also taking care of the high-level process control. Just a few examples. This is how the process is built in the SAMI process editor. Your own process can have manual tasks, it can have scheduler methods, it can have single liquid handler methods, it can have incubations and data queries. If you start new plates, you can start them in a calendar. The system schedules new tasks on the calendar, and it begins the first task, perhaps, for example, seeding cells. If you start new plates, you do the same thing as you did before on the calendar, but the system now looks at conflicts and warns of weekend evening requirements. For example, conflicts would be if you're overloading an incubator, or if you want to run the same task, let's say, on a device like a reader at the same time for different plates. And you got to solve these conflicts. You can solve these conflicts in the calendar. This is the calendar overview, interleaving multiple processes. Each process is indicated by a different color. It has a monthly overview and it has a daily overview. And again, you can move things around. So very often you have incubation times of a day or two, but it doesn't really matter to the hour. So just by simply moving a task a little bit, a couple of hours ahead or behind, very often does already help you to solve a conflict. Also in cell culture, it is very important that each individual well does count. Classic automation control software is designed to handle pieces of labware. In cell culture, every cell line, which is represented by a well, may be different. Different growth rates, different media supplement requirements, expansion splitting needs. And SAMI process management does exactly that. It controls the process by the well. As we've heard before, Biomek automation software has data handling in its DNA. So we can now use this data in the database, the data sets that do describe the properties of each individual well. We can use them now online to control method execution. And SAMI process management does introduce the concept of well groups. It is a virtual entity, multiple wells that do share a property or multiple properties that are on a single piece of labware or on multiple different pieces of labware can be defined as a well group. And then wells can be added and removed from this well group throughout the process. The tasks are then actually executed on this well group and no longer on a piece of labware. So that means, for example, to define these well groups, you can query in SAMI process management the database. So you can ask for properties, all types of properties that are stored in the database, right? So I give you an example. You have, for example, 5 6-well plates with 30 different clones. You read the confluency after 3 days. 1/3 of the wells spread across all these plates do exceed a threshold that you have defined for splitting. SAMI process management will now create a new well group, split the clones that are due, and incubate the others further. You will repeat the same procedure the next day, and the wells that are growing beyond the threshold overnight, and only those, will now be split. In summary, automating laboratory processes can help to increase throughput and reproducibility. It can help to remove human error, reduce hands-on time, and allow to work more efficiently. As you have seen, and this is just an introduction, Beckman Coulter does offer a wide range of hardware, software, and integrations and applications to enable successful and fast implementation of automated solutions in your lab. What we have seen that all current field of life science laboratory research have shown in the past to benefit from applying automated approaches with respect to speed and to data quality. The key to success is more in smart ways to control automation via software and develop and support methods as the hardware itself, because the hardware technology these days is very mature across the market. And not to forget, if you are using multiple samples, integrated data handling as part of the automated process is fundamental to generate reliable results in all areas of application. And with that, I thank the audience for their attention dialing in today, and I'm opening up for any questions. I'm looking forward to answer these.
Thank you, Dr. Overkamp, for that informative presentation. I would like to give our audience a quick reminder on how to submit questions. Simply type them in the Q&A box found by clicking on the green Q&A button at the lower left of the presentation window. We will answer as many questions as time permits. Okay, Dr. Overkamp, our first question is, accurate pipetting is important to all liquid handling tasks. How does Beckman address this, and how complex is it for the user to handle? Does it require scripting?
That's a very good question. Absolutely. I mean, besides everything that I've presented today, I mean, there is the fundament of very accurate pipetting that every liquid handling system needs to provide that's used to automate processes and generate reliable data. The way we handle that in Biomek software is we have something we call pipetting templates. And these pipetting templates are basically something a user can easily define just as writing a method, so it's icon-driven. And all operations that define the pipetting, like the move of the pipetting tip in the well, the actual pipetting speed, delays, etc., everything that you would automatically do manually, your brain adapts the pipetting operation to the pipetting context. All of that is in the pipetting template. And that definitely goes beyond liquid types. We have liquid types. But we can make these pipetting templates specific for any operation if we want, or for any context, such as a volume, such as a sample type, such as a labware type, etc., etc. And it does not require any scripting.
Dr. Overkamp, do you work with Illumina and others for new kit automated method development?
Yes, we do. So we are a partner to Illumina and others like NEB or Swift or Rubicon, and we have joint efforts. So basically, we are helping them to, when new kits come out or established kits are, we are helping them with our own experienced application support to come up with new methods, automated methods very quickly. And we also develop methods in-house and generate data, which are then validated with these vendors. And we also receive input from their side, what is important and what are the little details that need to be taken care of to get good results with their kits and chemistries. We're open, so we're not exclusive to anyone, and we will be in the future as well. And if you are looking at the full list of methods that we have already automated on our systems, you can see that really there is a pretty significant variety of vendors that we're cooperating with.
How do you manage sterility or containment in cellular workflows?
Yeah, cellular workflows, sterility, again, is a big point, a big concern for a lot of our customers. And there's two different approaches, basically. It all depends on the actual size of the instrumentation you are using. So if we're down to a very streamlined, simple B4K that's used for some basic liquid handling tasks in a cellular process, you can probably put that in a classical BSL-2 laminar flow hood. That's small enough. For larger systems, we do actually cooperate with manufacturers that do build custom enclosures, and there are different variations available. So sterility, we're actually offering HEPA enclosures ourselves, but if you want a real class 100 with a certification, we would cooperate with manufacturers of such enclosures. It also allows us to go one step further into biological safety, so we can offer BSL-2 or even full contained BSL-3 enclosures or containments that do allow the users to work with infectious particles in these systems as well.
Dr. Overkamp, more than the confluency, is it possible to count cell in a well?
Yes, that is possible to count cells in a well. Again, it's not so much a feature of the automation. It is more a feature of the integrated device itself. So if the device offers that option, we can definitely do that in an automated fashion as well. And on top of that, what we very often do if we need to do the cell counting is if it's adherent cells, we detach them and we use a cell counter. So we apply a little aliquot to a cell counter, such as the Beckman Coulter Y cell, and then do a cell counting and calculate the total cell number or concentration of cells per volume in that well.
Can you automate transfection?
Yes, we have a lot of examples where we have successfully automated transfection, and there's different ways of transfection. So we have automated transfection using chemicals. So transfection reagents. We have used biological transfection with viruses, and we have also used electroporation by integrating electroporators. So all different ways. The efficiency is usually very, very good. It does benefit from the reproducibility you get with the liquid handling automated across a plate. So there's multiple of examples where this has worked very well, and there are publications out there, scientific publications, not Beckman Coulter application notes, where you can find that in infectious disease. We have studies at the Max Planck Institute in Berlin for infection disease, which have conducted major screens on siRNA, and the transfection rates that they got there are published in those papers.
To elaborate on that question, we have an audience member that says we would like to automate the transfection process and also the core cell culture facility where we handle multiple cell lines. Could you provide more information on that?
Yes, absolutely. Probably not in this Q&A session because it's really down to the detail. So if that user is willing to give their contact details, we'll definitely be in contact with them, because the first step to a successful automation really is a very good understanding of that process, and again, in all detail, because in the end, it is the detail that will make the difference between a successful automation and not. So high level, yes, we can do that. I don't think the Q&A session now is the right format for that. Please, that user, I would really appreciate if you provide contact details, and we get back to you. We'll sit down and evaluate exactly what you need to automate, and we will come up with a solution. We'll be able to have some references for you that show what we've done today, and then we can take it a step further.
Thank you so much, Dr. Overkamp. We appreciate this great Q&A session. Do you have any final comments for our audience?
No, I don't have any final comments. I think I enjoyed it. I hope everybody has seen that laboratory automation really is can be a key tool to be more efficient in science of today. The request, I mean, the science environment has changed, right? So there is more and more data are required these days to be able to give a scientific publication. These data need to be very robust. They need to be handled with statistical approaches for data analysis, et cetera, et cetera. All of that only makes sense if you can generate data that you can trust, that are reliable, and laboratory automation is definitely a key tool to enable the generation of such data.
Thank you again, Dr. Overkamp. I'd like to thank the audience for joining us today and for their interesting questions. And a big thank you to our sponsor, Beckman Coulter Life Sciences, for underwriting today's educational webcast. This webcast can be viewed on demand through May 22nd, 2017. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now. We thank you for joining us, and we hope to see you next time. Goodbye.
Goodbye.